What's up, guys? My name is Ethan Polsana, and I just wanted to do a quick run through an explanation of a protein called RNA polymerase. So, RNA polymerase plays an essential role in gene expression, right? So, what is its function in gene expression? Well, it catalyzes a process called transcription. Excuse me. So here we see RNA polymerase in its natural setting, going to work on this strand of DNA, and basically what it does is converts this DNA into an mRNA, right? And we can see it converting here from a 5' end to 3' end, the new strand. The template strand is running from a 3' to 5' end. Here we can see the unwounding of the DNA double helix as polymerase goes to work. So what is this process called in gene expression? It's called transcription. Now, what is transcription? Excuse me. As we stated before, transcription is basically, it's a first part of the central dogma of molecular biology, and it's, the conversion of DNA to RNA. Through its catalyst, RNA polymerase. So transcription happens through 3 stages. The first one being initiation, the second stage elongation, and then the final stage termination. Now, the first stage of initiation is basically the beginning of transcription. It occurs when the enzyme of RNA polymerase basically binds to a gene called the promoter. Now, when it does this, it signals the DNA to unwind, so that the enzyme can go through and read the bases in one of the DNA strands. And basically, when it does that, it's preparing to basically create a new strand of mRNA with a complementary sequence of bases. Okay, so when it's done that, when it's ready, it goes into elongation, the stage of elongation, the second stage of transcription. Now, when it does this, it's basically, this is a stage where the addition of nucleotides to the mRNA strand, occur, right? So the RNA polymerase goes through, reaches the unwound DNA strand, and it starts to build the mRNA molecule. Okay, so it uses the complementary base pairs to do this. Now, there is a brief time during this process when a newly formed RNA is bound to unwound DNA. Now, during this process, adenine is in the DNA is bounded or paired with uracil in the RNA. Now, this is kind of different compared to DNA replication, where adenine binds to thymine. But here in this scenario, thymine is replaced by uracil, so they bind together. Right, and now after this, following this is the final stage called termination. Now, termination is the ending of transcription, hence the name termination. And basically it's given this name because when RNA polymerase crosses a stop, also known as termination, it sequences in the sequence of the gene, the mRNA strand is complete, right? It just tells it to stop, it's complete, and then it detaches from the DNA. So that's, in a brief sense, transcription and what RNA polymerase does in the process of transcription, working as a catalyst, doing all these things at once. Pretty amazing, guys. Of course, like every living thing, we're all made out of subunits, components. Same for RNA polymerase, all right? And the subunits include beta prime subunit, the beta subunit, alpha 1, alpha 2 subunits, and the W subunit, sigma. Now, I wanted to go through this real quick so you can see RNA polymerase and its subunits. Now, real quick, I just wanted to label everything. The beta prime is colored red, and the beta subunit is colored yellow, I believe, and alpha 1 is colored my apologies, just my screen was acting up. But anyway, I wanted to go through it again. My apologies for all that. So like I said before, the beta prime is colored red. The beta subunit is colored wheat, a light yellowish, as I said before. Now the alpha 1, the alpha subunit consists of 2 regions, alpha 1 and alpha 2. You can see alpha 1 in the, I believe the orange, if you could see that. And alpha 2 is yellow. And the sigma, the W subunit is in pink. So it's very, very, the pink is there, but it's the smaller subunit, which we'll get into in a little bit. So red, as we can see, it takes up most of the molecule, most of this enzyme, and the red is the, as I stated, the beta prime subunit. And the beta prime subunit is the largest subunit. And it contains part of the active center responsible for RNA synthesis and contains some of the determinants for non-sequence specific interactions with DNA and nascent RNA. Okay, and so it contains some of those components. Now the beta subunit, not the beta prime, but the beta subunit contains the rest of those components, right? So it basically is the second largest, as we can see through the color, and it contains the rest of the active center responsible for RNA synthesis and contains the rest of the determinants for non-sequence specific interactions with DNA and then RNA. Now, going to the alpha subunit, as we said, there's two regions, right? And it's the third, excuse me, and it's the third largest subunit. And it can basically these two the domains are the N-terminal domain and the C-terminal domain, right? And the N-terminal domain contains determinants for assembly of the RNA, and the C-terminal domain contains determinants for the interactions with promoter DNA, right? And basically, this type of promoter DNA, it makes non-sequence, non-specific interactions at most promoters and sequence-specific interactions at upstream element containing promoters, and contains determinants for interactions and factors that revolve around regulatory systems, stuff like that. Now the sigma W unit is the smallest unit. As we can see, the pink is there, but it's very very small. And basically what this unit does is facilitates assembly of RNA and stabilizes assembled RNA polymerases, right? So all these things work together to make the component of RNA polymerase subunits. I want to thank you guys for sitting through with this with me and being patient with me and just understanding the process of RNA and all the things that it contributes to. So real quick, so RNA polymerase, its function is to catalyze a process called transcription. And transcription happens in 3 stages, which is initiation, elongation, and termination, right? And all these things are done through the catalyst of RNA polymerase. And RNA polymerase itself is a hardworking protein because of these subunits, the beta prime subunit, the beta subunit, the alpha subunit with its 2 regions, and the W subunit depicted in pink, the very small pink. So thank you guys for sticking with me and just letting me go through these aspects I put into my protein paper. Thanks guys, and see you next time.